Imagine a technology that could do more than just treat diseases. Imagine one that could literally delete them from your DNA. We're not talking about an upgrade here. We're talking about a revolution in how we see life itself. Welcome to the world of CRISPR. So let me ask you this. What if a genetic disorder, something passed down through your family for generations, could just be erased? I don't mean treated or managed. I mean completely, permanently, deleted from the code that makes you, you. That is the insane promise we're going to explore right now. The easiest way to think about it is this. Your DNA is a giant instruction manual, right? 3 billion letters long. Now imagine there's a single typo in that book. Just one tiny mistake that causes a devastating disease. CRISPR technology is basically the ultimate spell check. It goes in, finds that one wrong letter, and fixes it. Simple as that. Okay, so the official name is CRISPR-Cas9. But honestly, just picture a pair of tiny, super precise molecular scissors. A little guide molecule leads these scissors right to the exact spot in your DNA that's messed up. And once it finds it, snip. It makes a clean cut. And that lets us do all kinds of things. Turn off a bad gene, cut it out, or even paste in the correct code. A tool this powerful sounds like the ultimate cheat code for biology, right? A miracle cure for basically any disease that's written in our DNA. The hype is, well, it's huge. But is it really that easy? I mean, the potential is just staggering. There are something like 6,000 to 10,000 diseases caused by just a single genetic mistake. These are called monogenic diseases. And in theory, CRISPR could target every single one of them. But, as you're about to find out, the theory is a very, very long way from reality. And here it is, the first big plot twist. According to experts like Dr. Terry New, using those little molecular scissors to actually cut the DNA, that's not even the hard part. The real challenge, the thing that keeps scientists up at night, is everything else. So, time for our first reality check, picking the right target. Before you can fix a typo in the code of life, you have to be absolutely certain you found the right one. And trust me, in our DNA, that is way more complicated than it sounds. The simple idea, the one we all kind of get, is that one bad gene causes one disease. So you cut out the bad gene, you cure the disease. Easy. But the reality is so much more complex. A single gene isn't a solo act. It's part of this massive, tangled network. It doesn't just do one thing, it can affect hundreds of other things in your body that we're just now starting to understand. Our genes are total multitaskers. Take the FTO gene. You might want to edit it to lower your risk of obesity, but surprise, it also helps repair your DNA. What happens if you mess with that? Or check out the APO4 gene, which is a big risk factor for Alzheimer's. You'd think getting rid of it is a no-brainer, right? Well, in young people, it might actually help with memory. So you see, editing a gene isn't like fixing a typo in a book. It's like knocking out a load-bearing wall in a skyscraper. Alright, next reality check, the delivery problem. So let's say you've picked the perfect gene to fix. Great. Now, how do you get your molecular scissors into the trillions of cells inside a human body that need it? So scientists basically have two main game plans. The first is called ex vivo, which just means outside the body. You take a patient's cells out, fix them in a lab, and then put them back in. The other plan, which is way more ambitious, is in vivo, inside the body, where you try to send the gene editing tool straight into the patient to do the job on its own. Now, even the simple ex vivo way is a huge deal. You've got to get the right cells out of the patient, ship them on ice to a super high-tech lab, do the incredibly delicate editing, and then get those fragile new cells all the way back into the patient's body without anything going wrong. It's a massive scientific and medical mission. But what if you can't take the cells out? Like, say, in the brain, or the heart, or the eyes? That's where the in vivo plan comes in. And this sounds like it's straight out of a movie. Scientists literally take a virus, strip out all the bad stuff that makes you sick, and then they pack it full of the CRISPR machinery. They basically hijack the virus and turn it into a tiny, microscopic delivery drone for a genetic cure. But this is where things get really, really risky. As Dr. Newman says, there are a lot of hopefulies here. You inject this viral drone into a person, and you just have to hope. Hopefully it finds the right organ. Hopefully it gets inside the right cells. Hopefully it makes the right edit. And you really, really hope the patient's immune system doesn't freak out and launch a massive, potentially fatal attack. Okay, our last big scientific reality check. This is the one that worries everyone. What happens if those molecular scissors are precise but not perfect? What happens when they slip? It's called an off-target effect. Basically, the CRISPR system makes a cut in the wrong place. Instead of fixing that one typo on page 500 of your genetic book, it makes a random cut on page 2000. And making a random, permanent change to someone's DNA? Yeah, that's something regulators like the FDA take very, very seriously. And get this is pretty shocking. Scientists don't even completely agree on what an off-target effect is. Is it any unintended change at all, no matter how tiny? Or is there like an acceptable error rate? The debate's still going, but the FDA is crystal clear. If this stuff is going to be safe for people, we have to be able to find, measure, and understand every single one of these mistakes. So we've wrestled with the science, but the final, and maybe the biggest challenge, it isn't in the lab at all. It's out here, in the real world of money, business, and big ethical questions. This is the final boss. The hype around CRISPR has started a total gold rush. Billions, with a B, have been poured into these biotech companies. I mean, just look at the stock charts. The promise of a one-time cure for humanity's worst diseases has Wall Street going crazy. But if you actually dig into their financial reports, you see a totally different story. And these are direct quotes. These companies are burning through cash. For years, not a single gene-editing product was fully approved. The manufacturing is insanely hard, and there's no guarantee anyone will even want these radical new treatments. It's the definition of high risk, high reward. And all that risk leads to the toughest question of all. If and when these cures actually hit the market, they're going to be wildly expensive. We're talking potentially millions of dollars for one shot. So, who gets it? Is this just a cure for the rich? How do we, as a society, decide who gets a second chance at life? Okay, so after all of that, the tangled science, the delivery nightmares, the financial risks, the ethical landmines, you might be feeling a little down on this whole thing. But if you are, you'd be missing the most important part of the story. Because despite everything, the progress is real, and it is breathtaking. Patients with diseases like sickle cell who've lived in constant pain their entire lives are now basically symptom-free after getting these treatments in clinical trials. A one-time therapy is stopping a fatal disease in babies. People with a type of inherited blindness are getting their sight back. This is not science fiction anymore. This is happening right now. So, here we are, standing at the dawn of a completely new chapter in medicine. The challenges are enormous, yeah, but the first few successes are already changing lives forever. We have unlocked this incredible, almost godlike power. The question isn't, can we rewrite the code of life anymore? We can. The real question is, what are we going to do with it?